what are the experimental techniques that we use to investigate the rate of transpiration? And also, what are the factors that can affect it? So the setup, the experimental setup, and I believe there's a little bit of overlap between this and even your GCSE. So you kind of learned that in a more simple level in your GCSE. Setup is called a potometer. Poto in Greek means to drink. So effectively, you're measuring how quickly the plant is drinking the water. Now, let's kind of like break down the setup here. Why do we set it up like that, first of all? Like theoretically, we could be measuring the amount lost, the amount of water vapor lost from the leaves. But technically, this would be next to impossible. You'd have to somehow, because it comes out in very, very small droplets, you'd have to somehow collect it and then measure its volume. So it's much more straightforward to actually measure the amount of water being taken up. So how do we do that? Well, effectively, we replace the roots in the soil with just a direct water source. So we take a plant and we cut off just before the roots. We cut the shoot. We cut it underwater and we generally assemble the entire apparatus underwater because it's important that no air bubbles penetrate any part of this apparatus. We may also do other things such as, so it's important that we have no air bubbles here in any part. We also kind of like, sometimes you put a little bit of Vaseline wherever you've got connecting bits of tubes, again, to avoid any air bubbles getting in there. And we're going to see, because it's important that we have the one air bubble that we use. We also cut the shoot underwater so that no air bubbles enter the xylem vessels here, because again, it's important that we have a single column of water going up the xylem. We have here a reservoir, which is just used to top up water so that we can repeat the experiment many times. And we connect the whole setup to a sort of narrow tube here that has a ruler behind it, so I can measure the distance. I introduce a single air bubble here. That's why it's important I don't have any other air bubbles anywhere in my setup, because I just want the single air bubble here. As the plant is absorbing water through transpiration, this air bubble is going to move from right to left. I can record the distance that it travels, and I know the diameter of this tube, so I can also calculate the volume of water. So the 2 pi r squared times the distance to calculate the volume. Yeah, that's it. Another caution that I can do before I start my experiment is to pat dry the leaves before I start my experiment, to ensure that there are no leaves, to ensure that there's no water on them. What are the factors that I have to control? Like with most experiments, the control factors are going to be basically anything else that can affect the outcome of my experiment. So I use this entire setup to be able to investigate one factor at a time. This is how a proper experiment is set up. You only test the effect of one factor at a time. So what are the possible factors that can affect my rate of transpiration? Light intensity. The higher the light intensity, the higher my rate of transpiration, because the rate of photosynthesis is going to be faster. So therefore, the stomata are going to be open for longer to facilitate gas exchange, and therefore more water is going to be lost from the leaves by transpiration. Second one is the temperature. The higher the temperature, also the higher the rate of transpiration, because the rate of evaporation from the leaves, transpiration is going to be higher. The effect of humidity, relative humidity. So basically, the humidity just outside the leaves. The higher the humidity, the lower my transpiration rate. Why? Because there's not going to be as big a gradient of water vapor between the inside of the leaf in the spongy mesophyll spaces and the outside. So the lower the gradient, the slower the diffusion of water vapor out of the leaf. And finally, air movement. So the higher the air movement, the faster, again, my transpiration rate is going to be, because as soon as a water vapor diffuses out of the leaf, it's being blown away by the wind, effectively keeping that gradient, that concentration gradient of air vapor steep. So humidity makes it less steep, air movement makes it more steep. And finally, availability of water. So the more water plant has access to from its roots, the more it can allow faster transpiration to take place. Let's have a look at an example question. Quantitative data analysis question. So a student was using a potometer to investigate the impact of total leaf area on the rate of transpiration. His results are shown in the table. So number of leaves on plant that represent the total leaf area, obviously, right? And the mean rate of bubble movement on the potometer, of course. Describe and explain what is shown by the student's results. So again, before you guys have a go, let's look at, let's break down our context. So our context here is basically this entire table, right? The data. My command is that, my command words are, of course, describe and explain, right? Here's my command words. But how exactly will I utilize them here? Well, I'm going to use the figures to describe, for the describe portion, and I'm going to use my knowledge, my understanding of transpiration to give reasons. And then the direction is to focus on the results and giving the reasons from our own knowledge. So have a go at writing this answer. Okay. Let's look at the answer together. So first, the describe portion, right? And as we said before, for my describe portion, I'm going to describe the general trend of what I'm seeing, followed by quoting a set of values to justify the observation that I gave. So mark number one, as the rate of leaf leaves on the plant increases, the rate of bubble movement through the potometer also increases. So just by looking at the numbers here, I can see number of leaves increasing, rate of bubble increasing. Easy, right? As an observation. So that's my 1 mark. For example, just literally pick 2 data points and quote them. For example, with 0 leaves, the bubble moves at 6 units, but with 8 leaves, it moves at 95 units. That's it. Then the rate of transpiration increases with the number of leaves as there are more stomata. So now we go to the explain part. So now we're going to use our knowledge to explain why this happens. There are more stomata for water vapor to diffuse out of. Note, to diffuse, not to evaporate out of, right? And therefore the rate of water uptake by the roots is also increased. Because effectively the evaporation from the leaves drives the uptake of water, which is why a lot of times if a plant loses its leaves, it's not going to be able to absorb water from the soil. Okay,